Namaste everyone. I am Mythili, your zoology faculty from Infinity Learn. Here we go with the second lecture from Animal Kingdom, which is Phylum Porifera. As promised, we are going in the sequential order. We started off other day with the basis of classification. How do we classify different organisms? Yes, I am going to do a little bit of brush of the previous session as well in the next 2 slides of mine, and then we move on to the first non-chordate phylum, which is Porifera. Yes, stay tuned. We will simultaneously carry out our class 12 lectures as well. Yes, I started off with human reproduction. Even those lectures will go on parallelly. As of now, this is the session from Animal Kingdom, which is the second topic, the lecture 2, which is Phylum Porifera, right? So from now on, as in when we are moving on to the Animal Kingdom, I thought let's do some of the amazing facts, okay, about these beautiful creatures, right? Let's do amazing facts instead of the regular quotes that I usually do. So I will start off with an amazing fact with the poriferans are commonly called sponges. They are around for a whopping 500 million years and are oftenly found in the deep sea. Unbelievable, right? So these are the organisms which have been around since millions of years ago. We did not, in fact, even know that these are animals. For a long time, these weren't even considered so. Only off the shore of California, they have found these organisms for the first time, and of course, in the coral reefs later in Australia. And that's how SpongeBob and SquarePants has come into the limelight. Most of you, or at least I should say maybe all of you, have known or do know SpongeBob, right? So the popular character that's based on our sponges, right? So what is that animal nature which allowed it to move to be moved to the Animal Kingdom? All that we are going to get there shortly. So here you see a very beautiful brightly colored sponge there from the picture. Yes, they exist in bright beautiful colors down there in the deep sea. Yes, they are very toxic also as well for various animals, predators, and that's how they're strong survivors, right? So this is the slide I was talking about, where I will brush off a little bit of what we have done in the previous session. So the kingdom Animalia, or the Metazoa, which is the subkingdom which is classified into Parazoa and Eumetazoa. Parazoa, the sidelined one, the branched one. What is the only phylum, did I say, under the Parazoa, which is Phylum Porifera. That is what we are going to precisely discuss now, Phylum Porifera, right? What did I say about you? If at all you recall from a previous session, these are the true. Wherever you come across this prefix EU, which means true, these are the true metazoans, which are further classified into radiata and bilateria, or diploblastic and triploblastic organisms. What are radially symmetrical organisms? You cut these organisms into 2 equal halves as long as the plane of division is passing through the central point. Bilateral symmetry, only one plane of division through the central axis is dividing them into 2 halves, or the mirror images, or the anti-mirrors. Diploblastic organisms, they have only 2 primary germ layers, ecto and endo. Meso comes into the picture, it becomes triploblastic organisms, 3 primary germ layers. Further down, bilateria is divided into the protostomia and the deuterostomia. So during that embryonic development, the pore that is observed, right? So this is what is referred to as the blastopore. Whether this blastopore is developing into the mouth first, proto meaning first, stoma meaning opening, just like what you have learnt in your botany part, stomata. These are the openings, right? So, protostomia, the blastopore develops into the mouth first. In the protostomia, it is developing into mouth secondarily. Later, the mouth develops, anus develops first in the deuterostomia. So, under protostomes, we have the acoelomate platyhelminthes. What did I tell you about A coming in the beginning? Absence. So, no coelom at all, absence of coelom. Pseudocoelomates, coelom is present, but it is not lined by the mesodermal layer. What is the only phylum under that? Aschelminthes and Schizocoelomates, a coelom that is formed by the splitting of the mesodermal layer. It's a true coelom, but the way it is formed by splitting of the mesoderm here. It is something that is formed from the outpouching of the gut wall there in the enterocoelom. Echinodermata, hemichordata and chordates, the most advanced phyla are all there. So, Parazoa it is, Porifera it is, that is what we are about to discuss today. So, when we look at ancestry, where did the sponges even come from? What we call in biological terms are the connecting links. So, connecting links are those group of organisms which possess the characteristics of two different groups, right. So, the protozoans and poriferans are connected by these proterospongia. What you see here is a beautiful picture that's given by our graphic artist, which is an example connecting the protozoans, right, and the poriferans. It's the proterospongia, choanoflagellate. You can call, we are talking about choanocytes as well, cells that are present within the poriferans shortly. So, proterospongia is the connecting link between protozoa and porifera. It comes handy when you study evolution in your 12th class as well. Connecting links are very, very important for you to understand the evolutionary biology, how different groups are connected or a certain organism possesses characteristics of two different groups. They are called the connecting links. Easy to remember, link is something that connects, right. So, the choanoflagellates or the proterospongia that you see here is the connecting link between protozoa and sponges. Within the name itself, easy to remember, protozoa, sponges, proterospongia, that is how you can remember that name easily. So, here is another beautiful picture of what we just studied, the classification part shown in a nice tree branches, how it is branching out, right. Look at this parazoa, which is separated from the rest, which basically did not evolve into any, or it belonged to the cellular level of organization, if you remember, right. And there goes all other beautiful creatures which are in the mainstream falling under the Eumetazoan subkingdom, right. So, porifera, why the name? All these non-chordate phyla, a lot of info is given from the very name itself, as and when I emphasize this most of the time, again stays true for all these names of all the non-chordate phyla. Porifera, from the very pori is something that you are understanding, it is pores or the openings. Straight NCERT diagram taken here, see, look at all these black spots that are present all over these organisms. What are these? Pores. What does fera mean? Bearing. Pore bearing organisms, and that's why under the phylum Porifera, right. So, why are all these organisms, or the major characteristic of these organisms that are grouped under Porifera is that they bear pores all over the body. So, it is like numerous mouthlets that are present throughout the body, right. Imagine ourselves with too many mouths all over the body, something like an alien, right. So, that is how these organisms are. There are numerous openings that are present throughout the body. That is how we could establish their animal nature, how they are taking in the water through these pores and letting out the waste after consuming what's important for them. That's how we understood they have actually got an animal nature. They have a certain canal system which helps them with feeding, and that is how they came into the Animalia group, right. So, members of this phylum are commonly referred to as sponges, something that we have already discussed. They are primitive multicellular animals. What did I say is the most common characteristic or something that should be there? Why most common? Actually, I take that back. It's in a character that should be possessed by all those organisms to be grouped under Animalia is multicellularity, right. So, multicellular organisms, these are the primitive multicellular organisms. What about their habitat? Again, the sound of the word is something, I am not saying this is a new word, obviously this is something that you would have learnt from your primary classes itself. To be remembering it very easily, habitat is nothing but address. If at all you would have learnt in your regional medium, right, languages, you should be able to very easily remember it as address, the place where you live in, that is the habitat. Can we say all the sponges are completely marine? No, there are freshwater ones as well. So, when I say aquatic organisms, all those live in the water. Yes, sponges are aquatic organisms, but do all of them live only in the seas? Are they all marine? No, there are freshwater sponges as well, like the spongilla that we are discussing, right, freshwater sponge. So, there are marine sponges, those which live in that deep sea, the one example I have quoted as the amazing fact you saw in the beginning, and there are ones which live in the freshwater as well. So, when it comes to their habitat, they are aquatic, yes, but they live both in sea as well as in the freshwater as well. Let us look at their habit, sessile. Now, when you go through all these 11th and 12th, you know this word sounds very familiar to you, immediately you come up with an answer. For now, it may sound fancy, basically sessile is not moving. These are not moving. The organisms which do not move are what are referred to as sessile, right. So, these are stuck in a place. Yes, their larval form is free swimming. During their developmental stage, the larva is actually free swimming, is able to move from a place to place, but once it becomes an adult, it's settling down in one particular place. This is one reason why initially they doubted whether it should be included in animals, because it wasn't moving anywhere like the plants, it is growing stuck in a place. So, these are sessile organisms, but as I said, when you look at their larval stage like amphiblastula, coeloblastula, parenchyma, these are all the larval stages we have in different organisms. These are the free swimming larval forms, they freely swim around. On the other hand, these are sessile, not moving, right. Asymmetry, if at all you recall from my previous session, now you know asymmetry means absence of symmetry. There is no symmetry, A is standing for absence there again. So, asymmetry, why asymmetry? Look at this particular organism, right. Try cutting it into two equal halves by any plane of division. Can you do it? No. So, these are asymmetrical organisms. Again, we cannot say all of them are, mostly asymmetrical. So, please do remember, when we say asymmetrical, these are mostly asymmetrical because some of them are radial, some of them do exhibit radial symmetry, but mostly these are organisms which cannot be cut into two equal halves in any which ways possible, and so they are referred to as asymmetrical organisms, right. Cellular level of organization, we did learn about levels of organization. What are the levels of organization? Let us recall quickly. Cellular, cells form tissues, tissue grade of organization, tissues from organ, organ level of organization, organs connect to form organ system. So, organ system level of organization. These are the four. levels of organization, of which sponges belong to the cellular grade. Again the same question I did explain previously also, let me put it forth again. It is a multicellular organism, right? Why not include it under tissue? Because tissue is basically a group of cells. Here there is no coordination, this division of labor. Look at this beautiful diagram here. These are the choanocytes which are performing a certain function where they attract water inside. These are the pinacocytes on the outer body wall, right? These are the porocytes which allow the water inside. In between there are also amoebocytes, the archaeocytes, the thesocytes. So there are different types of cells performing different functions. There seems to be a division of labor among the cells. If you look at the tissue definition, they should be, a group of cells which are of similar origin, right, similar type of extracellular material and doing a very specific function. So this belongs to the cellular grade of organization. What better picture can we put where you see all these different kinds of cells, right, within its body. So the body wall. The body wall of sponges has an outer pinacoderm. So the outer wall is what is called pinacoderm, the inner choanoderm, right, and the mid mesohyl. It's not even a proper layer, it's basically a cellular jelly like substance that's present in between. Meso stands for that middle, right, mesohyl. This is the jelly like substance matrix that's present between the outer and inner body wall, right. Some of the zoologists want to call this also kind of a diploblastic because it has two different layers, but many wouldn't agree. There's no proper primary germ layer formation that is there. But once it was asked question previously that in a way it can be called so because it has got these two layers, the outer pinacoderm and the inner choanoderm, right. So when you look at this outer layer, there is these flattened cells called the pinacocytes. And if you look at that inner, what are the choanoderm is mostly formed by these collared flagellated cells which are referred to as choanocytes. This is very, very, very important, can be your NEET MCQ. Collared flagellated cells which beat in order for the water to be attracted inwardly through the ostia, through those many mouthlets that are present. These are the choanocytes. And right in between you also see these thorn like things. What are these? These are the spicules. They form the skeleton of the sponges, which is also one of the reason why most of the predators do not like to eat them. They have these thorny structures present all over the body called the spicules. And they also got another very, very important cells called the archaeocytes. Why am I mentioning that these are important? These are the totipotent cells. So what do we mean by totipotency? The cells which can differentiate into any other cells of the body. So these are the ones which can actually differentiate into any other cells that I just mentioned. They can be pinacocytes, they can be choanocytes, they can be porocytes, they can be thesocytes, sclerocytes, amoebocytes. So these are the cells which can differentiate into any other cell possible in the body of sponges, which is why we are referring to them as totipotent. Totipotency basically means they can differentiate into any other cell. So archaeocytes are also quite important cells that you need to remember. Yes, of course, other cells are not mentioned in the NCERT, which is why I have not put it down there in the PPT. Otherwise, sclerocytes are the cells which actually form these spicules. There are spongocytes which form the spongin fibers, right? There are thesocytes which are actually storing the food. It's the old syllabus where you would have learnt about these thesocytes or the trophocytes or the sclerocytes or the amoebocytes. So these are all the other different types of cells that are present in the sponges. But most importantly, you remember, inner layer choanoderm is composed of collared flagellated cells called the choanocytes. The outer flattened cells are the pinacocytes, right? So please do remember these at least. So the skeleton, as I was saying, is quite thorny. There are these spicules that are present. What are these spicules? They're composed of either calcium or silica. It may be made up of either calcium or silica. Calcareous spicules or siliceous spicules. That is how we are talking about them based on what is composing these spicules. So the spicules are the ones which form the skeleton. There are other organisms also within sponges which are made up of spongin fibers along with the combination of certain siliceous spicules. So basically they may either contain these thorny structure spicules or very minute fibers which actually form that soft. Have you come across this sponge at your home? Very silly question, right? All of you know what a sponge is, like the cleaning one. Yes, of course, nowadays we get a lot of synthetic ones. Only what you see at your homes is mostly that synthetic one, but it's actually originally made from these organisms. That's why these are referred to as the sponges. The spongin fibers have this great retency of the water, and that's how these usage of sponges came into the picture. Now of course most of the things that you see are synthetic ones, not the originally made from these organisms. Again, we would end up extincting these organisms as well if at all we are exploiting for that reason, right? As we did with most of the organisms, unfortunately. So yeah. So if at all you see these spicules which are made up of 3 of the thorn-like structures, it's a triaxon. You see there is a diactinal monoaxon. There are 4 of them, pointy structures which is tetraxon, monoaxon, amphidus. Spongin fibers look something like that. So trying to give you a picture of how these spicules or spongin fibers look like, right? So canal system, very, 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 very important. If at all you take all these previous question papers, this is something that you will see most of the time. Canal system is the very core essence of the sponges. This is what is allowing major functions, whether they have to take in food, whether they have to help with removal of the waste, whether it is about the respiration, or in fact reproduction as well. We are going to look at it, how it helps. So what is canal system? There are these minute pores, as I told, throughout the body. What are these referred to as? Ostia, the plural form. Ostium is singular. So water goes in. Look at that beautiful line that is running through. What is the central cavity called? Spongocele. What is the cavity within these organisms called? Cele is cavity, spongo organisms with the cavity within sponges is spongocele. Nice, easy to remember, right? And it has one major exit through which the water goes out, which is called osculum. What is that particular opening which allows the water out? It is called the osculum. In fact, that is also nicely regulated by certain cells which are present here, which are called the myocytes, little contractile cells which actually help with closing and opening of osculum. So why is water going inside, coming out? That's how it is getting its food. It's a suspend feeder, it's a filter feeder. It literally filter out all the food that it requires and lets out the waste. So it is helping with excretion as well. It's getting its oxygen, so respiratory gas exchange as well. In fact, it helps with cross fertilization as well. Remember this aspect because the final, just penultimate among non-chordate phyla, we are learning about Echinodermata, where also you would see a water vascular system, a little different from the canal system, but mostly there's a comparison and the questions can be asked. So nutrition, very important. Your NCERT line says it's an intracellular digestion. The digestion is basically taking place within the cell. Intra is within. Within the cell there is digestion that is taking place. Look at this beautiful picture again, how the food goes in, how the cells, the archaeocytes or the choanocytes which are taking in that food, it's a microphagous one, it's a filter feeder, it's a suspension feeder where it takes in all the food and allows the waste to go out. It's a, what kind of a digestion is it? Intracellular digestion. Later in Nidaria we will see both intra as well as extracellular digestion. For now, sponges have intracellular digestion. How about the respiration and excretion? No specific respiratory structure. Look at these are not very advanced organisms. The general body surface only helps with exchange of the gases. And if at all we are talking about its waste, it's an ammonotelic. What does that mean? It's excreting ammonia. It is excreting ammonia. So what do we call these organisms? Ammonotelic organisms, right? Asexual reproduction, quite fascinating experiment that was done. In fact, it was nicely grounded in a mortar and a pestle, and then it is finely filtered through a cloth. And what about all these cells that have obtained from the sponge? Each of them can give rise to another sponge. What a nice thing that you observe. High regeneration capacity. It has got a very high regeneration capacity. That is how it's represented pictorially. Look at how many pieces it is done, but still it's able to reproduce or grow into a new sponge, right? So that is a type of reproduction, a sexual one, which is called fragmentation. Fragment it and it develops into a new sponge. Budding. So this is also something that you have learnt in your school level itself, how a small bud grows up into a whole new organism, and you know it is becoming again sessile, choosing another surface where the bud breaks off, attaches itself to the new surface and grows to a new thing. So far asexual reproduction, one is fragmentation, two is budding. Gemule formation, again a very nice thing that this organism does. During unfavorable condition, these forms these little gemmules. These gemmules are nothing but nicely filled up with what cells? Archaeocytes. And it is also guarded from the predators because of all these spicules around. And there is a small opening called the micropyle. When the favorable conditions return, all these archaeocytes come out through these micropyle and they form new sponges. What a beautiful mechanism, right? So what are all the things that we have learned in asexual reproduction? It is fragmentation, it's budding, and it is gemmule formation, right? And you must be wondering, how does canal system help with sexual reproduction, cross fertilization? There are these sperms that are formed in one sponge. Obviously, there is water that is, traveling through the ostia, through the spongocele into the osculum. So, through that the water, the sperm actually arrives here and it fertilizes with the egg cell that is within the mesohyl, right? And the zygote gives rise to the free swimming larva. It swims outside and develops into a new sponge. So that is how the canal system is actually helping with the sexual reproduction in these organisms. There are these different larval stages based on the different types of sponges. We have coeloblastula, stereoblastula, amphiblastula and parenchyma, right? So these are the important examples. As I always say, animal kingdom, please learn your examples, scientific names. Scypha, Sycon, Spongilla, freshwater sponge, very important. Euspongia, bath sponge. This is the sponge that I was talking about, how you could daily use at home, the sponge that you are very aware of, right? Although you have not seen this deep sea creature, the sponge that you have seen at home, it's basically this, the Euspongia. Classification is something that's not included as part of NCERT anymore, but just for your info, it has got calcarea, meaning it has calcareous spicules, as simple as that. Sycon and Leucosolenia are the examples. Hexactinellida, it's siliceous spicules. Why the name Hexactinellida? It forms these spicules which are, of 6 rays like this, which is why the term hexa, 6, hexactinilidia. The siliceous spicules can form these nice transparent glass sponges. What do we have there? Euplectilla and hyalonema. And then demospongia, it has got lot of spongin fibers, spongilla and euspongia. Here I should give a special mention of Euplectilla. It is something that's also referred to as Japanese wedding gift. Why do you think this is given such fancy term, the Japanese wedding gift? Did you observe that sponge before, how there is sperm that's traveling? Similarly, larvae of some other organisms, for example, a certain species of these prawns, they get inside. Because they are getting their food and shelter, they stay there. Once they grow big, they cannot get out of the sponge. Osculum is too small to allow these organisms outside. So they are there together forever, right? That's the caption behind the marriages everywhere in the world, which is why it's a precious, precious wedding gift that's given to the couple in Japan, which is why it's referred to as Japanese wedding gift, also called the Venus flower basket. That's a little of story for you along with the poriferans, right? So let's get to the quiz, the 5 questions there. Pores all over the body of the sponges. What are the pores called? Ostia, right? Singular being ostium. Describe functions of canal system. How does canal system help? It helps in nutrition, providing with food, respiration, provision of exchange of gases, excretion, removal of waste, also cross fertilization. Collar-like flagellated cells are those cells which are collar-like flagellated. What are those? Those are the choanocytes. Sponges undergo sexual reproduction by, they undergo asexual reproduction by fragmentation, right? Gemule formation, right? Budding. Skeleton in sponges is made of, it could be either the siliceous or the calcareous spicules. Apart from that, it can be also made up of what? Spongin fibers, right? So there we have the answers, and there we also come to the end of beautiful world of the sponges topic, right? Next up, from the animal kingdom, we are going to discuss the coelenterates or the cnidarians. At the same time, from human reproduction also we will move ahead with the lecture 3 simultaneously there as well. So keep studying, keep learning, because learning is infinite and fun, and I want you to become all those world-class doctors there getting through your NEET examination, right? Keep learning.